ರೆಡಿ ಇದೆ ಸರ್. 2 ಮಿನಿಟ್ಸ್ಗಿಂತ ಮುಂಚೆ ಬಂತು ಸರ್. ನನಗೆ ಕರೆಕ್ಟ್ ಬಂತು. ಹೆಸರಿಂದ ಸುರೇಶ್ ಅಂತ. ಸುರೇಶ್
ಹಾಂ ಸರ್. ಹಲೋ ಸರ್.
ಮಾರ್ನಿಂಗ್ ಸರ್.
ಸರ್ ನೀವು ಕಳಿಸಿದ್ದೀನಿ ವಾಟ್ಸಪ್ನಲ್ಲಿ. ಕಳಿಸ್ತೀನಿ. ನಮ್ಮ ವಾಟ್ಸಪ್ನಲ್ಲಿ ಕನೆಕ್ಟ್ ಮಾಡ್ಕೊಂಡು ನೆಕ್ಸ್ಟ್ ಟೈಮ್. ನಿಮ್ಮ ಪ್ರಿನ್ಸಿಪಲ್ ಅದಕ್ಕೆ ಕಳಿಸ್ತಾ. ಪ್ರಿನ್ಸಿಪಲ್ ಹೆಸರು. ಹಲೋ ಸರ್.
ಗುಡ್ ಮಾರ್ನಿಂಗ್ ಸರ್. ನೀವು ಇಲ್ಲೇ ಬರ್ತೀರಾ ಸರ್? ಮಾತಾಡ್ತೀರಿ? ಇಲ್ಲಿ ಬರ್ತೀರಾ? ಬನ್ನಿ ಮತ್ತೆ. ಫಸ್ಟ್ ಅಡ್ಮಿಟ್ ಬೇರೆ ಕೊಡಬೇಕಲ್ಲ, ಅದರಲ್ಲೇ ಮಾತಾಡೋದು.
I give the instructions. You will welcome. Then you will welcome and you will give the vote of thanks. In between, if it is there, I will like question.
ನಾನು ಯಾಕೆ ಬರ್ತಿದ್ದೀನಿ ಗೊತ್ತಿಲ್ಲ.
So ma'am, you will do the welcome.
ವೆಲ್ಕಮ್ ಅಲ್ಲ, ಪ್ರೆಸಿಡೆಂಟ್ಸು
ವೆಲ್ಕಮ್ ಯಾಕೆ ಬೇರೆ ನೋಡಿಲ್ಲ. ನಾನು ವೆಲ್ಕಮ್ ಮಾಡಬಾರ್ದು. ನಾನು ಜನರಲ್ಲಾಗಿ ವೆಲ್ಕಮ್ ಅಂತ ಹೇಳ್ತೀವಿ. ನಾನು ವೆಲ್ಕಮ್ ಅಂತ ಮಾಡಬಾರ್ದು. ಎರಡು ನಾನೇ ಮಾಡ್ದಂಗೆ ಆಗುತ್ತೆ. ಜನರೇಟ್ ಹಾಕೋದು
ಇನ್ಸ್ಟ್ರಕ್ಷನ್ ಜೊತೆ ವೆಲ್ಕಮ್ ಮಾಡಿಬಿಡಿ ಅಷ್ಟೇ.
ಪ್ರೆಸಿಡೆಂಟ್, ಸೆಕ್ರೆಟರಿ, ಪ್ರಿನ್ಸಿಪಲ್, ಮಂಜುನಾಥ್.
ನೆಕ್ಸ್ಟ್
Good morning everyone. I am Arpita, Assistant Professor at Sarada Vilas College. I welcome all of you to this webinar. Please kindly go through the following instructions. Please mute your audio throughout the webinar. Turn off your camera and mic during the time of the webinar. Be careful not to click on the option present now because it may interrupt the speaker. Please use the chat box for any questions or communication during the webinar. As soon as you enter the meeting, please enter your details, name and institution name in the chat box. The feedback will be shared with you in the last part of the webinar. So please kindly submit the feedbacks, and e-certificate will be made only for the participants who have given the feedback. Please kindly cooperate in conducting this webinar successfully. Now I would like to welcome our President, Shri B.S. Parthasarathy, who is always supporting for all our endeavors. And also I would like to welcome Honorable Secretary H.K. Srinath. I would like to welcome our Principal, Dr. Devika M. And also I would like to welcome Dr. Manjunath. I would like to welcome today's speaker to the webinar, Dr. M.G. Suresh. I welcome you, sir.
Hello.
Hello, sir.
Hello.
So it's audible, sir? Hello?
Hello.
Sir, am I audible, sir?
Hello.
Sir, am I audible?
Hello. Hello.
Hello, sir.
ನಾನು ಯಾರಿಗೆ ಫಿಲ್ ಮಾಡಬೇಕಾಗಿಲ್ಲ ಡೈರೆಕ್ಟ್ಲಿ
Hello, sir.
ಅವ್ರು ಕೇಳ್ತಾ ಇಲ್ವಾ ಫೋನೇ ಮಾಡಲಿ ಅವ್ರಿಗೊಂದು ಕಾಲ್ ಮಾಡಿ ಸರ್.
Sir, call.
ಹಲೋ.
ಹಲೋ.
Hello, sir.
ಹಾಂ ಸರ್, ನಾನು ಲಾಗಿನ್ ಆಗಿದ್ದೀನಿ.
ಓಕೆ ಓಕೆ.
ನೋಡಿ ಸರ್,
ಹಾಂ
ಮೇಡಮ್ ಇದ್ದಾರಲ್ಲೇ?
ಹಾಂ ಇದ್ದಾರೆ ಅದೇ ಚೆಕ್ ಮಾಡ್ತಾ ಇದ್ದಾರೆ, ಒಂದು ನಿಮಿಷ.
ಹಾಂ,
ಲಾಗಿನ್ ಆಗಿದ್ದಾರೆ, ನೀವು ಸ್ಕ್ರೀನ್ ಕೊಡ್ತೀನಿ ಅಂದರೆ ಕೊಡಿ.
ನಾನು ಆಡಿಬಲ್ ಇದ್ದೇನೆ ನೋಡಿ.
ಓಕೆ ಓಕೆ. ವಾಯ್ಸ್ ಇಸ್ ಆಡಿಬಲ್ ಸರ್.
ವಿಡಿಯೋ ಆನ್ ಮಾಡಿದ್ದೀನಿ ಈಗ.
ಎಸ್ ಅಲ್ಲ. ಹಾಂ ನಾನು ಮಾಡ್ತೀನಿ ಮೇಡಮ್.
Sorry for the sorry for the technical interruption. Now I request Manjunath, HOD and the Assistant Professor Microbiology Department, to introduce the speaker webinar, today's speaker to the webinar.
ಹಲೋ.
Hello, good morning everybody. It is a great pleasure for me to introduce and welcome the speaker of this present morning, Mr. Suresh MG. This person merely requires any introduction, though Sir was born on 12th August 1975. He completed MSc in microbiology, qualified SLET exam and is pursuing PhD. Mr. Suresha is currently assistant professor in Government College, Hassan. He has guided more than 50 scientific models for about more than 100 students, published technical writings and cartoons in various Kannada newspapers and magazines. Sir has received many awards for his great talent and achievements. He received international recognition for guiding second PUC student on topic. NPV, a farmer's friend from Intel, which was selected among top 30 for Young Scientist Award during 11-12. National level guidance for high school students for participating in Inspire Award competition at IIT Delhi on topic Starlight Avatar during 15-16. State level guidance for BSc students for achieving second prize in topic Star Avatar 15-16. State level guidance for BSc students for achieving first prize on topic organic farming during 12 and 13. He has also conducted extracurricular activities related to subjects like Feel Employable, a career guidance to BSc students, microbiology in daily life, killer diseases, cancer, medicals, personal hygiene, myths and facts about science. organic farming, solid waste management, sustainable agriculture. Other than this science, he is also very interested and good at different kinds of art like resume preparation, interview facing tips, calligraphy, cancer counseling, scientific models, graphology, cartooning, paper quilling, stage decoration, and event management. Thermocol craft, pencil, organic, Varley art, abstract art, vegetable carving, doodling, etc. Sir, conducted group activity sessions to improve one's personification, such as be a winner, how to be a better learner, concentrate, confidence, and achievers, brainstorming and creativity, make your own milestone, mock interview, etc. Thus, without any further delay, I would like to call upon Mr. Suresha MG to share his knowledge with us. And also, our principal speak some words on this webinar session.
A very good morning.
A very good morning to one and all, and a warm welcome to the delegates of this webinar on industrially important microorganisms organized by the Department of Microbiology as a part of the webinar series of our Sarada Vilas College. At the outset, I would like to thank our Honorary President, Mr. K. Sarada, and Secretary, Mr. H.K. Srinath, for their encouragement and support in conducting this type of event. Next, I would like to express my sincere gratitude to Mr. Suresha M.G., Assistant Professor of Microbiology from Government Science College, Mysore, for accepting the invitation and delivering this talk on industrially important microorganisms. I hope this webinar will definitely help students and faculty of microbiology, which is focusing on industrial microbiology. I congratulate Mr. Ari, Head of the Department of Microbiology, for taking interest in organizing this webinar. I will be failing in my duties if I forget to mention the names of members of our technical team, Dr. Jagdish Krishna and Ms. Arpita, for making all the technical arrangements in conducting this webinar. I request all the participants to make use of the expertise of the speaker and get full benefit out of this webinar. Thank you, one and all. Jai Hind.
Thank you for all the well wishes. Now I request our speaker, Mr. Suresh MG, to take over the session.
Hello.
Hello, sir.
Good morning, all. Madam, am I audible to you?
Yes, sir, it's audible, sir.
Video is clear? My video is clear, madam.
Yes, sir. Yes, sir.
Okay. Good morning, all. First of all, I want to thank Sarada Vilas Institution, the patrons, B.S. Parthasarathi, President, H.K. Srinath, Secretary, Principal, Dr. M. Devika, HOD of Microbiology, Professor R.A. Manjunath, and staff of Microbiology. Also, for giving technical help, Dr. Jagdish Krishna as well as Arpita. First of all, this presentation, whatever that I am presenting, is by taking the experience of myself in the industry about 10 years. I worked in production department, quality control, research and development, marketing, as well as sales and HR. This presentation, that is industrially important microorganisms, will be concentrating more towards students' perspective, where students, they should equip themselves to to get the candidature for the industries, and how they can learn the techniques in their BSc as well as in MSc, so that they can do good job when they get into any fermentation industry. And now I want to present the topic. Yeah, industrially important microorganisms. As we know, microorganisms are friends as well as foe to us. Means they bring about fermentation, they bring about the products which are required for humankind as well as for the nature, even for the agriculture and all. But somehow they are also like foe to us, like enemies. They are they cause the diseases to us, and we are prone to that. Apart from that, if we are talking about industrially important microorganisms, we should know that industrial microbiology, which is a branch of biotechnology that applies microbial sciences to create industrial products in mass quantities, often using microbial cell factories. These are multiple ways to manipulate the microorganism in order to increase maximum productivity. Suppose if we take an organism as industrially important, then what we had to do is, first we should know that whether this product is fit for the industry. If it is not fit, we have to improve it. There are certain techniques where we can improve those by strain improvement, like adaptation techniques, so that it can give maximum yield. The manipulation of organisms in order to yield a specific product has many applications to real world, like the production of some antibiotics, vitamins, enzymes, amino acids, solvents, alcohol, dairy products, etc. So microorganisms play a big role in the industry with multiple ways to be used. Medicinally, microbes can be used for creating antibiotics in order to treat the diseases. So if you come to the history, as the production of the products by using microorganisms, like as early as 6000 BC, the Babylonian and Sumerian used yeast to make alcohol. Very surprisingly, alcohol was produced in the early years, so that the people actually didn't know whether the products are coming because of the microorganisms. Like unknowingly, they came to have the alcohol, and even the baking industry started. After that, Pasteur developed a new way of approach to industrial microbiology, where the Pasteur, he solved the problem of souring of wine and spirits, where one of his associate, he came and addressed him, saying that his wine industries are suffering because of the sour smell, and the wine lovers are not tasting it good. So then what he did, he went on searching the problem, and he came to know that there is lactic acid fermentation by lactic acid bacteria, that it yielded lactic acid rather than alcohol. So then he suggested that the must which is prepared should be heated up to 72 degrees centigrade up to 20 minutes, so that it can kill those. So that is the pasteurization technique. So this is because of this pasteurization technique, even till we are following that in dairy industry and other food beverages. And during the First World War, the British scientist Chain Weizmann developed a fermenter for the production of acetone by using Clostridium acetobutyricum. So this is the fermented product where the acetone was prepared by using the microorganism. In 1950, Hindustan Private Antibiotic Limited, Pune, made the first pilot fermenter in India. Actually, making the fermenter is challenging, where we had to do the we had to provide the regulated environment to the microorganisms so that we can grow them in mass, that is in more number. However, the use of microorganisms to produce industrial chemicals started only about 100 years ago. So the developmental phase in industrial microbiology is relatively ancient, involves fermentation of alcoholic beverages and bread, large-scale cultivation of yeast cell of brewing and baking. Actually, now there are more than like 25,000 liters, 50,000 liters, like this. The fermenters are coming up, and they are used extensively now throughout the day, like in the 3 shifts, where it can be utilized in the full form to give the to yield the microorganisms. Industries known as fermentation industry introduce fermenter and production medium. Yeah, production medium is different. As we say, we grow microorganisms in our lab by using basic medium. But in the production medium, they will tweak the composition of the medium so that it can influence the growth of microorganism as well as product recovery. So in this, there are they introduced different fermentation techniques, that is batch culture as well as the continuous culture, and even some other type of fermentations which will suit that particular product and the microorganism. So use of microorganisms as bulk sources of protein for animal and human food is called SCP. Yeah, single cell proteins, where spirulina and chlorella, they are used. Even for the photofermentation, the fermenter is different. Especially in improved countries, they use photofermenters where the glass tubes are used as the continuous system. The continuous media will be flowing and even the cells will be harvested. So in the biotechnology, the recombinant DNA technology has brought more boon to the production, as we can just manipulate the gene to improve the quality. So why we have to take the microorganisms? Microorganisms are the mini chemical factory. They possess a broad variety of enzymes. They have high metabolic activity, so it can grow and multiply rapidly. They possess a large surface area for the bulk, for the quick absorption of nutrients, and they can grow on inexpensive substrate. Byproduct of this may be the substrate of another. So they are amenable to genetic manipulation. I want to put an example where when I was working on trichoderma production as well as BT production, there is like we used to have the raw materials which are nothing but the byproducts. Sometimes the agriculture waste, even the agriculture waste which are rich in cellulose, that can be used for the production of these microorganisms. Even sometimes if you get the byproducts, that can be used as a substrate for some other organism. And these can be genetically manipulated so that we can get the desired whatever the product, or maybe the concentration or yield of the product can be increased. So first we'll come to the fermentation. Fermentation is a metabolic process that produces chemical changes in organic substance through the action of enzymes. Actually, fermentation has many meaning. People initially, it was quoted like fermentation is an anaerobic process carried by microorganisms to produce the desired products. And later we said fermentation, any action carried out by microorganism is fermentation. As far as industrial microbiology is concerned, this fermentation is carried out maybe aerobically or anaerobically to produce the desired product. So in biochemistry, it is narrowly defined as the extraction of energy from carbohydrates in the absence of oxygen. In the context of food production, it more broadly refers to any process in which the activity of microorganisms brings about. That's what I told initially. The science of fermentation is known as zymology because of the zymase enzyme. So these are all some fermentation tanks which are initially used, as well as in industry, these are all used much. The baby fermenter that we can say is around 55 liters to 50 liters, which is used as a pilot scale that can also be done. But here, these are all the huge fermenters. They have much of like regulated environment, and also the traditional method is also followed. One thing I want to highlight for the students is like
ಇರುವ ವ್ಯಕ್ತಿಯು ನಿಮ್ಮ ಕರೆಯನ್ನು ಹೋಲ್ಡ್ನಲ್ಲಿ ಇಟ್ಟಿದ್ದಾರೆ.
when we for students, especially nowadays in industries, these fermenters are regulated online on the software. That's why it is suggested that if they are very well versed with AutoCAD and all, it's very easy for them to operate online. Otherwise, in particular industries, they have designed their own software where these kind of regulation can be monitored. Like if the pH has to be varied, sometimes we have to add some alkali or acid. Initially, what we used to do is manually we used to fill the like columns or pipes so that whatever the amount of the buffers are required, we used to dispense it. But now the things are changed. That is hello. But now the things are changed. That is
ನೋಡಿ ತಿರ್ಗಾ ವಿಜಯ
yeah, now the things are changed. Like the fermenters are monitored online in computer software, and whatever the regulation can be done is done online. If you come to the structure of fermenter, I think already final year students, they have studied this. I'm not going to be going in detail in this. Otherwise, I want to just highlight some tips on fermentation. If anybody goes into the fermentation industry, there are some tips and tricks where we can adopt ourselves to get the desired product and also to increase the yield of the product. Actually, this is the structure of fermenter, where you can find the baffles and the impellers. The main thing is this area, head space, is very important. Around up to 75% people fill the media and 25% is left. When we are fermenting the gram-negative bacterium, we have to take care that it should be lesser than 75%. It should be around 60%, because these contains lipids, and these lipids are going to accumulate as foam. And beating the foam, there are mechanical beaters, and also we can use some anti-foaming agents like silicone oil and other oils. So that is more important when it comes to the industrial fermentations. Otherwise, what happens is, suppose after the reactions, these will Yeah, hope I resumed the presentation. So what happens in the industry? It is very critical in maintenance of the fermenter, especially when it comes to like between two fermentations. The cleaning of the fermenter is really a task where we have to clean the fermenter without a single cell is being inside the vessel. So that is also more important. We should give more prominence to the cleaning of fermenters before starting the new inoculation when it comes to batch fermentations. Yeah, the main component of the fermenters are like vessel, as we know, it is made up of the metal, especially the stainless steel, and there are impellers. Impellers helps in proper mixing and heat transfer, and especially in submerged fermentation, they play a very important role. And there will be sparger. Sparger is also helpful for mixing, as well as it will introduce the required gas, especially oxygen, into the medium. And baffles, they will increase the efficiency of aeration, and also there are probes like temperature control, pH control, dissolved oxygen, everything can be measured online, and there will be inoculation port, feed port, and sampling port, as well as delivery port. So when it comes to the fermentation stage or steps, we have to select the fermenter, what kind of fermenter that we need. The fermenter is, suppose if it is a solid state fermentation, the fermenter will be like flat bed. Otherwise, if it is like submerged fermentation, the fermenter will be, the height of the fermenter will be more. And we have to select the microbial strain, whatever that we are using. Microbial strain is also very important. Whatever the strain that we use should be like industrially improved. It should be industrially improved, so that already I told, the yield as well as the adaptation to the particular fermentation technique is very important. And the fermenter media, this varies from companies to company. We initially we use in our lab like basically nutrient agar, potato dextrose agar, or maybe Zepedox agar. But if you go to the production media or industrial medium, the concentration of these may vary according to the product, desired product, whatever that we have to yield. So fermented process or fermentation process, this is again very important, that whether we are going for batch fermentation, continuous fermentation, or fed batch fermentation, so that that particular organism will fit into that or not. Sometimes what we have to do is we have to do trial and error, maybe 3 or 4 fermentations, where we can just standardize the incubation period, and sometimes we have to induce certain products, especially secondary metabolites. And in case of Bacillus thuringiensis, we have to induce for formation. In case of penicillin fermentations, we should see that the penicillin production is there, that penicillin production should be started. For that, some additives will be added. Like that, that is the upstream process. Upstream process is the entire process. We can get the maximum attainance of the cells, microbial cells. Still, the maximum load is attained, we should go for fermentation. The downstream processing is nothing but, once we get the desired organism along with the broth or medium, we should downstream, means we should purify the microbial cells. So there are different techniques, precipitation, centrifugation, filtration, all those things. So that will be there. So after that, we have to do the quality control or quality assurance. Quality control is that where in the end product, or maybe in the fermented broth, we should get certain number of cells, and it should be devoid of pathogens like Salmonella, Shigella, as well as the total contaminant load should not increase, or maybe no any contamination should be there. So production is very important when it comes to the product recovery. Whatever after the broth, the fermented broth is attaining the maximum load of microorganisms, the product can be recovered. So then comes to the packaging. Packaging is depends upon the type of product. They use sometimes tin, sometimes the HDPE, high density polyethylene, and after that it will go to the marketing, and before that simultaneously it will go to quality control. So there are types of fermentations already I mentioned: solid state fermentation, liquid state fermentation. There are batch fermentation, head batch fermentation, continuous fermentation, surface fermentation, and submerged fermentation. As suppose if you are growing fungi in industry, especially the filamentous fungi, it's very difficult because we have to use the solid surface, and to get the solid surface, the fermenter has to be like flatbed. Sometimes people will design their own kind of vessels where they'll get the maximum surface area. Yeah. Now already I told, if we have to say when the organism is fit for industrial production, there will be some improvement. We have to improve that particular strain. Suppose if we isolate a particular strain, that can be improved. That particular strain can be improved. That particular strain can be improved by certain techniques. Like by inducing the mutations, we can improve it. Sometimes even we can starve the organisms so that we can just grow them in different kind of environment so that it can adapt to those conditions. As well as we can also just get the product recovery. Means suppose if we are desiring some secondary metabolites, the growth period will be varied. Like we can minimize the lag phase, we can increase the log phase, we can stabilize the stationary phase, like that. So suppose if you are growing fungi and we need only the conidiospores of the fungi, what we can do is we can minimize the myceliation period. So for that, the carbohydrate concentration has to be increased. So that if you increase the carbohydrate concentration and decrease the nitrogen concentration, it will induce the spores, and that can be harvested easily if we are only concerned about the conidia or conidiospores. So this, as I told, E. coli strain K12, it is the improved strain, and this strains of like strain improvement, it is more important in industry because it will save the time, and it will save the cost, and even it will save the manpower. So ideal characteristic of a strain is it should grow rapidly. Yeah, once we initially isolate that organism, it may be growing standardly in the normal growth phase, but we can induce those rapid growth by inducing the mutations, sometimes by varying the composition of the media and also incubation parameters. So genetic stability has to be there. Suppose if a strain gets improved, its genetic stability has to be stable. Otherwise, it should not change its genetic composition or makeup, so it can act differently later. So then, no toxicity to humans. There should not be any toxicity to humans. That is already we know. And ability to use cheaper substances. Yeah, sometimes some microorganisms, they need specialized chemicals. Suppose if we, for the same source, if you find any like byproducts or any products which are low in cost, that will be easier. Then elimination of production of compounds that may interfere with downstream processing. Sometimes in the downstream processing, what happens? Suppose we are extracting only the spores. There will be some others like media ingredients, and suppose if you don't need the mycelial fragments and all, that will be intervening. So we should design, or we should just improve the strain so that the desired product is more in that. Then to improve the use of carbon and nitrogen sources, as the CN ratio or the carbon and nitrogen, if they use more and if it directly proportional to the yield of the yield, then it will be good for us. Then reduction of cultivation cost. Yeah, this is the critical thing the industrial people, they will be looking on. Every year they will be always asking you,
Madam, Madam.
Hello.
sir. Yes, sir.
Madam, this my screen is visible now?
Yes, visible.
My screen, my presentation is visible?
Yes, sir, it's visible now.
Okay. Because here and there I'm getting the pop-up I'm no more presenting. That's why. Please let me know if there is any interruption.
Yes, sir. Sorry for the interruption. Please, I request all the participants not to click on the present now option. Please pin your presentation, like Suresh MG, there is a presentation slide. There are two slides with the Suresh MG. So Suresh MG presentation, please pin that slide and keep it like that only. Please don't click on the present now option.
One request for all the participants, please don't click on the present now option, and you can pin Suresh MG where I am presenting, so that there is no any hindrance in the presentation. This is my request. Okay. When it comes to, shall I resume, madam?
Yes.
Hello. Sir, shall I resume? Shall I continue?
Yes, continue, sir.
When it comes to the reduction of the cultivation cost, it is more important in the industry. If you go to any industry, what they'll do is they'll ask us to cut the cost of raw materials and fermentation process. This is very critical. So what we have to do is always we have to keep on trying different raw materials, so that will be fit into our industry or particular type of fermentation. And also the next important aspect is the shorter fermentation time. Suppose we say for the fungal fermentation, sometimes we go up to 5 to 7 days. So if you reduce at least 1 day, the cost of production will come down, because for 1 day we will spend more on electricity, more on water, more on manpower, and even the production efficiency of that particular industry, it will come down. So if we reduce the fermentation time, if we get 5 days harvest or 4 days harvest, then it will increase the production efficiency as well as the yield, total yield. So how we approach for strain improvement? As I told, strain improvement can be achieved by mutation. There are some physical agents like UV. You can expose the organism to UV for certain seconds to minutes, so that it can change itself to produce the better product or desired product. Then even some chemicals are used like nitric oxide. Otherwise, we can go for recombination techniques, as we know, transformation technique, conjugation, as well as transduction. Apart from this, recombinant DNA
ಅಲ್ಲಿದೇನೋ ಡಿಸ್ಟರ್ಬೆನ್ಸ್.
lot of other products has been come on. So if you come to the food and dairy industry, as we know, the bakery industry, the bread is produced, and also the other bakery products are produced, like cake, wherever the fermentation has to take place. And then production of vinegar, soya sauce, product, cheese, and yogurt. And if you come to the pharmaceuticals, the main and foremost thing is the production of antibiotics, vaccine, vitamins, and enzymes. Then if you come to the alcoholic beverages, it's production of wine, beer, brandy, rum, whiskey, and other spirits. And in agriculture, it's production of biofertilizers, biopesticides. So other products are organic outputs, bioleaching, biofuels, etc. So if you come to the products like soya sauce, cheese, yogurt, beer, wine, bread, or that is, the glucose will be converted into respective products. So role of microorganisms in food and dairy industry. Microorganisms are involved in producing many foods and beverages, fermentation products, characteristic flavors, aromas, and consistencies of various foods. So microbial metabolism has other functions. They act as preservatives. They destroy many pathogens, microorganisms, and the toxins. They can also add nutrition value to form a vitamin and other nutrients. So microbes are used in food production. So these are all some microorganisms which are used in different products of food industry and dairy industry, where as we bread is produced by yeast Saccharomyces, and the substrate is wheat, rye, and other grains. The soya sauce is produced by Aspergillus oryzae, and the substrate is soybean or wheat. Again, the cheese is produced by lactic acid bacteria, and the substrate may be milk. And yogurt is produced by Streptococcus thermophilus, and the substrate is milk. Like sauerkraut, where lactic acid bacteria, it is going to convert the cabbage juices to sauerkraut. So in the food industry, as you know, bread making, that involves Saccharomyces cerevisiae. One thing I want to highlight here is this Saccharomyces strain is improved a lot now, when compared to initial. There are some industries who have improved this, especially for brewing industry. Even in baking industry also, the strain improvement of Saccharomyces is done extensive job. Hello?
Yes, sir, it's visible, sir. The screen is visible.
Because I got the message like you are no longer presenting.
No,
Somebody.
sir, that's fine, sir. Even though they click on present now, your PPT will be coming, sir.
Okay, no problem. Thank you. Yeah. Yeah. In the strain improvement, actually, especially people have studied more on Saccharomyces cerevisiae, especially ellipsoidesis, where it can bring more of alcohol production, like adaptability to certain environment, and there will be huge fermenters. And in that fermenter, the organism which are growing in the log phase, that have to be maintained constantly. So for that, strain improvement is done, and even in the baking industry also, for the leavening for the forming of these fermented products, this is like tweaked well, and the bacteria. Excuse me. Like in the dairy industry, in the cheese making, milk is treated with lactic acid, and also the bacteria, an enzyme called renin, that is particularly used for hydrolysis of the protein, and it will coagulate into the curds. And the liquid portion of the milk, that whey, is drained out. The whey is separated and from the curds, and the curds are aged, pressed, aged, and ripened to form the different kinds of cheese. So different microbes in the early and late stages of processing give rise to cheese in different characteristics that we have already studied. So in the dairy industry, cheese making plays an important role, because most of the cheese, whatever that the developed countries were using, has become their daily need in their food. And this is some process, as this is the pasteurization of that milk will be done. Then the starter culture is added, lactobacillus, and then it will be coagulating the milk. So after that, the whey is drained out, and whatever the solid part, it will be pressed and it will be kept for ripening. So role of microorganisms in pharmaceuticals. So more than 65% of all antibiotics are produced by microorganisms, especially actinomycetes, that is Streptomyces. One thing I want to highlight, like we used to hear in our village that some old lady or the old people, they don't get any infections, or they never go to hospitals, or even though if they get some kind of fever and all, they will manage themselves. The story behind, or maybe the idea behind this is they are always playing, or they are always doing work in the mud as well as in farmyard manure, that is cow dung. With cow dung, the farmyard manure is rich of Streptomyces, So as they'll be preparing the slurry of this farmyard manure and applying on their yard, sometimes some people they apply that onto the wall because they'll be living in some small huts. Naturally, the compounds which are adhered to their hands while preparing, it will bring them defense from the microorganisms. That's the story. So even actually we used to see like if anybody dies in a home, we used to sprinkle that cow urine all over the place in the home because that we say it is like decontamination. That's the decontamination process that initially we started because it contains a lot of volatile compounds like formaldehyde, glutaraldehyde, and all. That is the naturally we are using this as rituals. So that is a main source of antibiotics, that is streptomyces, which are present in soil as well as in farmyard manure. More than 65% of them are used in pharmaceutical industry. I want to highlight one thing. Microbiology students, biotechnology, biochemistry students, they get more of opportunity in pharmaceutical industries, and there are different sections like production, quality control, and research and development. If they get any chance in the production, quality control, and R&D, they should get into the job. And if they get directly some kind of like quality control job, it's very good. They should opt that. And if they go to production, they can work for maybe 1 or 2 years in the production. Then they can jump into the quality control. And even getting into the research and development, that's R&D, is very difficult unless they can assure you, like, or you can assure them like that you can do good job in the development of the products, then it is good. Otherwise, if you get any field, any section, any production, quality control, or R&D, you can jump in. So that is for the pharmaceutical industry. And as we know, vaccines are produced in large numbers, like DPT vaccine, diphtheria, pertussis, and tetanus, measles, mumps, and rubella. As we know, the Pasteur Institute of India, which is situated in Kunur, Tamil Nadu, they produce this in a large number, and it can be like disseminated all over country for the mass vaccination and all. And then vitamins, vitamin B12 is produced by Pseudomonas and Propionibacterium, and enzymes. As we know, streptokinase is a lifesaver. When anybody, like any person who comes sudden heart attack, to thin the blood, this streptokinase enzyme is introduced. So role of microorganisms in pharmaceuticals, as I told, antibiotics. Penicillin is the first antibiotic which is developed by Alexander Fleming, Florence Chain, and then streptomyces and even Bacillus species. There are plenty of microorganisms which are involved in the production of antibiotics. And alkaloids, that's Claviceps bacteria, and steroid transformations, that's Rhizopus and Arthrobacter. Insulin production, human growth hormone, somatotropin, interferons. So these are all produced by E. coli and yeast. So production of antibiotics. The study of antibiotics began by Alexander Fleming, as I already told, and initially it was started with Penicillium notatum. Now a lot of species have come. Lysozyme has been used, and then most of the penicillin are 6-amino penicillinic acid derivative. They contains the beta-lactam ring, which is responsible for antibacterial property. Streptomycin is an effective antibiotic against Mycobacterium. As we know, the penicillin production is by Penicillium chrysogenum. The spore suspensions are inoculated onto the wheat bran medium. Yeah, if you come to the present, this one, fermentations, wheat bran medium and other lactose medium are used for the penicillin production. The thing is agitated for 1 to 2 days. Agitation plays important role in the mycelial growth. The mycelial mat has to be formed, and then the pH is maintained over 7.2 to 7.3, and the temperature is 73 to 83 Fahrenheit, and 5 to 7 days of fermentation. Yeah, this varies, and there are 3 phases where initially there will be build-up of mycelial growth during the log phase, and then consumption of lactose. This mycelial mass
Hello, sir.
Or start produce, then at the end of the stationary phase, the concentration will come down. So that we have to determine and we have to harvest. Same thing happens in Bacillus, that is Bacillus thuringiensis fermentation, where we should induce the endospore formation. If you take only the vegetative cells and if you formulate and if you spray it on the field, they may not be more effective because they are lack of endospores. Otherwise, these vegetative cells has to go to environment where they suffer the optimal condition, then they can induce the endospores. The thing is, by that time, the pest, Helicoverpa armigera or Podaptera lupida, which are called as cotton ball worm and tobacco worm, they will get the upper hand and they will spoil the entire field. So that's why when the product is going into the market or into the field, it should be equipped with the main active ingredient. The main active ingredient here is the penicillin, and in case of Bacillus thuringiensis, there should be endospores which will release the triprotein. So, so using the rotary vacuum filter, penicillin is separated from fungal cell. That is the downstream processing. So fungal biomass is dried and used as animal feed. That is a byproduct. Then quality assurance, we'll check the the parameters which should satisfy for the packaging as well as marketing. So role of microorganisms in beverages. Yeah, this is very interesting. People are interested in this because of alcoholic beverages. Microorganisms are involved in the production of almost all alcoholic beverages. Alcoholic beverages are produced by fermenting malt and cured beer by beaver's yeast, that is Saccharomyces cerevisiae. Beer and wine are non-distilled products. They will not distill because the concentration of alcohol is around 7 to 12%, and the whiskey, brandy, plus some spirits like vodka, they will be concentrated by means of distillation. So when it comes to the alcoholic beverages, we are unknowingly, some people will consume the products which are partially fermented. In case of fruit, especially grapes, if we observe the bunch of grapes, like 1 or 2 grapes will be softened and they will taste different because naturally they contain a layer of yeast on their skin. So because of some accidental injury, they can get into the flesh and they can bring about the fermentation. Maybe the partial fermentation, the concentration of alcohol may be 1 or 2%, and if we taste that, it looks good for us. So it tastes better, and if it increases or maybe other pathogen comes, then it will be spoiled. Otherwise, especially in grapes, we can observe this. And initially, when we were studying, at that time, like there was some stories, there were some like practices which was mentioned is some Baba came from Himalaya and he is giving some kind of bread or roti which is having some kind of super medicine. Suppose if anybody consumes the like fermented or maybe that juice which is prepared by the roti, they will be like cured from almost all diseases. The thing is, the persons who used to be said as saints, what they used to do is they used to sit in a place, proper place, and saying that the like colony or maybe the expensive people saying that I have bought this roti from Himalaya and this is like miracle roti which will cure your diseases. And he used to instruct the followers saying that you should bring the tea decoction. Tea decoction means as we prepare tea by adding water, tea powder, sugar, everything, they should boil in a vessel, and white cloth has to be covered. That has to be brought to him so that he will do some puja and all. After that, they have to keep it in their home for 7 days. Initially, what he will do is he will take a piece of roti, whatever that he has, he will put it into the decoction, that tea decoction, and they should go and keep it near the God's place. They worship that for 7 days. After 7 days, they get the juice along with a new roti. So people or astonished to see that new roti had come from a piece of roti, whatever that person given. So if they consume that, whatever the liquid, they used to get relief from the pains, especially the knee pain and all. The story is like this. Actually, the fonda is like it is nothing but it is the alcohol which is produced from the glucose, because they have provided a sugar solution which is carbon source and a tea powder which is nitrogen source. As we know, yeast are ubiquitous; they present everywhere, and they will come into the solution, that decoction. They will start to ferment. The glucose is converted into the alcohol, and as we know, people who get the alcohol, they get relief from this mild pain and all. So that's the story, and people used to bluff by using these kind of techniques, that is microbial techniques. So as we know, the wine making process, people use red grapes. There are a lot of other fruits can be used. Sometimes it can be fortified with some malts and all, malt powder and all. But here we can see the red grapes or the purple grapes has been crushed, and the must is prepared. After that, the inoculum is added, that is yeast, and then it will be kept for incubation along with sugar and some other preservatives. So this wine is prepared, that red wine, white wine and all. We know different substrates are used, like some people use pomegranate, some people use some other like strawberry wine, all those things. Depends. Regionally, wherever the more raw materials are available, people will adopt for such kind of fermentation. In the wine making, it must undergo primary fermentation. Malolactic fermentation by bacteria is most convert malic acid into lactic acid. Yeah, that's one of the criteria that we have to think. And in the secondary fermentation, aging is more important in wine. So, and also after the final product, the blending and bottling is done. This is where the schematic representation, where the must is prepared by adding the grapes, and hope final year students, they have prepared the wine in their lab itself, and this fermentation will be taken place up to, I think, 21 days in our lab condition. Otherwise, in industrial conditions, it may come down also because of the improved strengths. And the clarification of the wine is done. It is kept for aging, and then it is bottled. Like that, beer fermentation is there, where the malt is used, and then inoculated with the Saccharomyces cerevisiae. After that, this fermentation, I think I will not go detail into that. I'll just skip this. This is steps where the malt is prepared, and the mashing kettle that will be kept, and then the enzymatic activity takes place. Some hops are removed, and the yeast culture is added. That yeast will vote the beer to form the beer. Again, that will be kept for filtering, aging, and all. So microorganisms in agriculture industry, it is more important nowadays, because we are going to replace that whatever the chemicals we are using, that is NPK, nitrogen, phosphorus, and potassium, in the form of urea, MOP, and DAP, diammonium phosphate, muriate of potash. So these are all the supplements which will give the macronutrients to the plants, that is nitrogen, phosphorus, and potassium. So if we add more of these, as we know, the soil becomes sterile, and also most of the concentration, whatever that we use, which will not be utilized by the plants. So the main alternative for this is biofertilizers. Biofertilizers are nothing but the microorganisms which can supplement the nitrogen and other nutrients. So as we know that rhizobium is produced in large numbers in mass production, especially final year students, they have a chapter for the production of rhizobium, Estobacter, Spirillum, Azolla, and VAM and all. In the rhizobium production, the rhizobium is isolated from the soil, and it can be fermented in EMA medium, that is yeast extract mannitol agar, and then that will be mixed in carrier material, especially lead, lignite, perlite, vermicompost, and all. That will be marketed in packets, and farmers will use them by seed implantation, by seed coating, and that seedling dip. Sometimes even they will broadcast. And the second thing is the biocontrol agent production, that is Bacillus thuringiensis, which is a bacterium, then Trichoderma fungi, NPV. See, you can think now for the production of like if you are producing in lab, this bacteria and fungus can be grown, but how this virus NPV, because they need a definite host. Yeah, since NPV means nuclear polyhedrosis viruses. There are CPV also. With nuclear polyhedrosis viruses, they will be grown only in living cells or maybe in animal cell line. So what we have to do to produce this NPV, we should rear the worms. We should rear the larva of Helicoverpa or Podoptera in large number. When they attain like 4th or maybe 5th instar, 4th or 5th instar, we should inoculate the diet, whatever that they are eating, with NPVs. So what they will do is NPV will be ingested into the digestive system. There they will be hydrolyzed, and that will kill the worms. After there is some significance visible symptoms, or that becomes very black in color, those who are infected. On plants and all, it will hang in inverted V shape actually. Otherwise, in multi-cavity trays where it is produced, actually I produced this. I produced NPV, Bacillus and Trichoderma, but this production of NPV is challenging. Like once we get that black colored larva, which is dead, that will be collected, that will be grounded, that will be grinded in the mixer. After that, it is purified, centrifuged, and the viral component will be harvested. That viral component will be formulated along with the blue and all, that is whatever that robin blue is available, and then it will be disseminated in the field, or in the they will be mixed in the water, diluted, that will be sprayed in the field. Then microbial herbicides like Coletotrichum, Dressera, these are all produced in large number so that they can control some weed. So this is the schematic representation of biofertilizer production. Yeah, one thing is very important. From the mother culture to slant, and slant to the working culture or maybe the broth, this plays very critical role. Means when it come to producing a potential microorganism, here there should not take any, there should not be any mutation, so that the mother culture is preserved well in a controlled environment. Maybe sometimes it is refrigerated. Otherwise, people will use frequent subculturing. Within 7 days, there is subculture. Again, they will keep on going with the subculture. This plays very critical role. From the broth, it will come to the small flask. Again, it will come to the rotary shaker. Sometimes here, baby fermenter will be there, 5 liters to 50 liters. Then it will go to actual fermenter. From the fermentation, that will be harvested and mixed with the carrier material. Then that will be kept for curing. Then it will be disseminated or dispatched to the farmers. Yeah, maintenance and preservation of this industrially important microorganisms is very crucial. Most of the people prefer preserving this material or maintaining this their product, their component, this one microorganism in periodic subculturing, as I told, because this will ensure the log phase organism growth. Organisms will be active, and whatever the metabolites which is going to be produced, like secondary metabolites and all, they will get more. So because of that periodic subculturing, because they will be in the same kind of environment, there is no any change of temperature, and overlaying with mineral oil for the longer periods they want to keep, they'll do it. Even sometimes sterile soil is added bacterial protect system where some beads are coated with bacteria and preserved. Cryopreservation, we know, in nitrogen, 70 degrees. And the lyophilization, the sublimation technique, where the water is evaporated and the cell is kept inactive. Yeah, when it comes to the conclusion, microorganisms have always been exceedingly useful to mankind, even when their existence was unknown. They will remain an essential part of many basic industrial process technologies. The development of recombinant DNA technology has further intensified interest in industrial microbiology by expanding the potential of new products and applications. The use of specialized microorganisms to produce such products on the industrial scale will probably become more important. As far as the industrially important microorganisms is concerned, now most of the industry is concentrating more on improving the strengths so that they can reduce the cost of production and all. Sometimes while improving the strength, we can also get negative results, like mutation can happen for good as well as for bad. Even we'll get some junk organisms, which is the maintenance will be very difficult. We have to just discard them, we have to sterilize them before we are discarding into the environment. So my presentation here it ends. Apart from that, I want to highlight few things on the industrial techniques where our students, like BSc students or MSc students, if they go for the interview, what kind of questions will be asked there? Dear students, please don't worry about the questions will be asked to the molecular level and all. The questions which are asked is basic level. So especially the technique, media preparation, it may be sterilization or even the maintenance. The pure culture techniques you should learn very well in the lab. So especially if you're inoculating an organism, it should be pure culture. It should be pure culture without any single organism. See, I will quote example here. Suppose this is the mother culture. We say there may be like 10 to the power of 8 cells per gram here. Okay. By the time it will reach here, like the main fermenter, it will be like trillions together, the cells. And these are all the series of steps it has to cross. So what happens is, suppose if you are not getting the pure culture here, and if you got any contaminant, and if you don't report to the authority, and as such if you start fermenting this, sir, we'll be using a lot of media here, we are using a lot of manpower, and all other production costs will increase. By the time it reaches here, it becomes not valuable. So there is no any value for that because it has contaminated. So make sure that you should not you should have techniques or maybe pure culture techniques in such a way that you will get the required or desired organism. The contaminant level and all, yeah, sometimes the contaminations may come because of the inoculation and all. Nowadays it is very reduced because most of the things are automized here. That is like automatic inoculation, that is machine inoculation, automated inoculation will be there. Initially what we used to do is, suppose if there is any like inoculation port here, we used to swab this area with 70% alcohol and keep 2 or 3 spirit lamps, and then we used to pour the inoculum. That was the traditional method, but nowadays it is not there. It is connected here. So what happened? Now it is connected here. It is connected to the automation where somewhere the media is, the inoculum is added, and it will go without any manpower. So that is the strategy. And one thing we teach here for the students regarding all the theoretical aspects as well as practical aspects. In industry, if you get into any industry, there will be like log maintenance, logbook maintenance. Suppose if you use any instrument like pH meter, incubator or laminar airflow, you should record there what the material that you used and for what purpose, and if you taken any pH, what is the pH, what is the sample, like that. In the QC, it plays very important role. That actually normally will not teach. And also you should know the SOPs. SOPs means standard operating procedures. even to open or close the refrigerator, there will be standard operating procedures. So this will be pasted. The company protocol number will be given, and that will be pasted on respective instruments or maybe near the area that will be displayed, and you have to follow that. Then second one is the log maintenance, and the third one is like there will be like a periodic auditing where people will come and check the media preparation, how much of media you have used, and what is the result of inference of your samples, whether they are positive, negative, false positive, all those things. That will be like seen there. So my suggestion to students is what you have to do is here you learn the basic techniques like media preparation and like pure culturing technique. And if this is the basic requirement for any industrial, they will not directly go and ask you regarding the PCR and all. They know on what condition you have studied. Only thing is, if the company is having the certain techniques, they will just train you. Initially, they may train you for 6 months, 1 year or so. And if you know or very well versed with the technique, they will absorb you in the same companies, and that will give very good package. So my suggestion to students is learn the theory and be like expertise, get the expertise in the practical applications, and that is enough for you. And moreover, if you are going to the industry and all, you should also learn some computer basic techniques. I prefer AutoCAD. It is good for operation of the fermentation and all. Otherwise, the basic Windows application is necessary nowadays. So by this, I will end my presentation. So these are all some companies, some industries which are involved in microbial fermentation. There are a lot of names, Dr. Reddy's and all. And even in the pharmaceutical industry is booming, like Cipla, Ranbaxy, Shanta Biotech, and AstraZeneca. These are all. No, AstraZeneca is involved in COVID vaccine production. Okay. Finally, I want to thank the patrons, B.S. Parthasarathi, President of SVEI, and E.K. H.K. Srinath, Secretary, and also Principal Dr. M. Devika Madam, and also HOD of Microbiology, Manjunath Sir, actually who gave me this opportunity. I'm very thankful to him, all the microbiology department staff, all the staff, and all the work who are directly or indirectly involved in this program. And finally, I want to extend my thanks to Jagdish Krishna and Arpita Madam for hosting this webinar and making it success. Thank you one and all. If anybody is having any clarification, they can ask me, especially the industrial part, like whatever takes place in the industry, the equipment process, and what will be in the lab. That if students are very interested to know, I can able to give the answer. And this is nothing but sharing of experience. That's all. That's all.
Thank you, sir. It was very much informative. Now I request if any of the participants have the questions, you can put it in the chat box. Otherwise, you can just unmute your camera and mic and you can ask the speaker. Anybody has a question? If anybody has a question, we can just move on. Otherwise, we shall proceed to the vote of thanks. Yeah, I just hope that there are no questions from the participant end. I would like to thank Suresh MV for those in his busy schedule who made a time for us and sharing. Hello? Hello? There are no questions in the chat box. So we shall end the presentation. So I would like to thank today's speaker Suresh MG. Even though in his busy schedule, he made a time for us and who shared an really informative presentation with us. And actually, the presentation was so informative. We are not expecting any questions from the participants. And few participants are asking for your PPT. If it is shareable, I'll just mail that if it's permittable, sir. Yeah, I would like to thank Suresh MG, and also I would like to thank Sri B.S. Parthasarathi, President Sarada Vilas Education Institutions, and also H.K. Srinath for a continuous support in conducting such webinars. And I would like to thank Dr. Devika, Principal Sarada Vilas College, who continuously encourages us in conducting such webinars. And also I would like to thank Dr. Manjunath as HOD and Assistant Professor, Department of Microbiology, who took the initiative in conducting this program. I thank you, sir. I would like to thank Jagdish Krishna, Head of the Department Computer Science. I thank you, sir. And I thank all the faculties who are coordinating in conducting this webinar. Last but not the least, I would like to thank all the participants without whom this program would not be successful. Thank you all. I'll be sharing the feedback link in the chat box. Please kindly fill up the feedback link so that I can just send you the e-certificates. Thank you one and all. Thank you, sir. Sir, you can exit the meeting, sir.
ಮೆಸೇಜ್ ಹಾಕ್ಬಿಡಿ 3 days
Yeah, the feedback link is shared in the chat box. Please the feedback link is shared in the chat box. Please kindly go through that and fill up the feedbacks. Thank you one and all.
ರಿಜಿಸ್ಟ್ರೇಷನ್ ಫೀಡ್ಬ್ಯಾಕ್ ಅಂತ ಬಂದ್ಬಿಡ್ತಾರೆ ಇದು ಸೆಷನ್ ಅಟೆಂಡ್ ಮಾಡಿದವರು ಫೀಡ್ಬ್ಯಾಕ್ ಜಾಸ್ತಿ ಅಲ್ಲ